Well, thanks for the kind introduction. And this will be now a switch. We are switching to mice, and the hope is that I can convince you that actually you can learn something from mice that maybe then will have really impact later also on humans. But before I start, I would like to thank Alan for selecting me for this prize and for the BBRF Foundation for giving it to me. It's a great honor for me to have received this. And as you have seen, Alan, he has been a pioneer in studying how affections during pregnancy increase the risk for offspring mental disorders. And we had a fantastic collaboration together where we used the mouse and tried to get a little bit at causality by looking if artificially activates the immune system during pregnancy in a controlled way, how this then will affect the development of the offspring, and then how this will be affecting brain function once these mice are growing up. And we made some very interesting observations how it affects cortex function and then cognitive behaviors that are affected in patients with schizophrenia. But I will not talk about this today. I will talk about a more recent story where we study also what brain circuits may be important for cognitive deficits. And just to remind you, if you think about schizophrenia, unfortunately, we still have to diagnose the disorder by its symptoms because there are no biomarkers. Now, you can't do a blood test. You cannot do a brain scan that tells you whether a person has schizophrenia or not. So we have to use still the symptoms. And we have the most characteristic symptoms, which are the positive symptoms, including disordered thought, hallucinations, and delusions. This is what everybody knows about schizophrenia. Someone is hearing voices, for example. But then there are other symptoms, like negative symptoms and cognitive deficits, which they are maybe not as known. They are not also necessarily specific for schizophrenia, but they can be very important for the long-term prognosis of the disorder. And this could be like social withdrawal, blunted affect, decrease in motivation, but also cognitive deficits in attention and something called working memory, which is basically the ability that you right now can listen to me, think about what I'm saying, and then maybe you ask a question, but then you can forget about this information. So this fluidity to do things is very important. And if you don't have this, it's very hard to function in an everyday life and to keep a job. And so therefore, we really have to do something to understand better cognitive processes like working memory. And although we have some medication that helps against the positive symptoms, dopamine D2 receptor blockers, they don't work in all patients. They have terrible side effects. But there's basically no medication so far that really can help you to improve your working memory. And this is something very important. So what people think is we have to study the basic mechanisms of this. And then if we understand them, then we may come up with better therapeutic approaches. So if you think about cognitive deficits, and if you think about working memory, the first structure in the brain that comes to mind is called the prefrontal cortex, which is like a cortical area just here in front of your brain. And numerous imaging studies in the last 30 years have shown that whenever you take a patient and engage this patient in a task, in let's say a working memory task, a cognitive task, you see an altered activation of the prefrontal cortex. But the problem is that the brain doesn't work that the prefrontal cortex is doing something. The brain is organized in circuits. And the circuitry that is involved in this cognitive task also includes the thalamus, which is a small structure in the middle of the brain. And whenever you now look in the studies, you see that there's an altered activation of the prefrontal cortex. You actually see that also the thalamus shows altered activation. And this may be because they have this reciprocal connectivity, so they're talking to each other. Now, the thalamus now is a small structure in the middle of the brain, where also there's indication that there may be actually some structural changes. So what we thought a couple of years ago is that a small damage there could have a big impact because the thalamus may actually influence how the cortex is working to regulate working memory. And so we basically asked a simple question whether if you now decrease activity in the thalamus, as you see in what has been observed in patients with schizophrenia, would this be sufficient to induce cognitive deficits that usually are thought to be mediated by the prefrontal cortex? And so in humans, this is very difficult to do, but you can do this in mice because we have the tools to do this. And you can now take a mouse, yeah, and use the virus where you express now a designer receptor, and you just inject this only in the medial dorsal thalamus, just as projections to the prefrontal cortex. And then this designer receptor, we know via intracellular signaling cascades that it can activate inhibitory potassium channels. And that means if you now have a mouse where this receptor is only expressed here, you can now inject the designer drug into the mouse that you only decrease the activity of neurons in this structure and nowhere else in the brain. And you can measure this. So if you now take an electrode, put it into the medial dorsal thalamus, and you have a mouse that is running around, you see that this neurons is firing at about 4 or 5 hertz, yeah? And if you now inject to the mouse this designer drug, you see that this activity is going down. And it's actually a relatively mild manipulation, surprisingly mild. If you now sample more of the cells, you see that in the medial dorsal thalamus, about 30% of neurons, they show a decreased activity by about 30%. So it's actually very mild manipulation, but you will see it has big effects on the behavior. And so how do we test working memory in the mice? They're not as smart as us, so you have to use more simple tests. And one very simple test is this T-maze. So in this T-maze, the mouse originally sits in the start arm, and then you randomly choose to go to, let's say, the left arms. No? And now the idea is, as I told you before, is that the mouse, the idea is has to keep some information online, yeah, and then use this information to solve the problem, in this case, in this trial, and then it can forget about it, and then you start the next trial. So in this case, now it has to learn or encode that during the sampling or encoding phase, I was left. Mouse will go back to the delay arm, will sit around here, but it has to remember I was here. And now during the choice phase, when we open both arms, the mouse has to go to the right arm. And then you can change the length of this delay and can make it harder. And therefore, the memory component of this working memory gets harder. And I have a video to show this, which was embedded, but then the mouse didn't move rightly, so we have to switch off, get out and get into the video. Just the mouse. So it just left the start arm, and the mouse is now collecting the reward. So we randomly choose this arm to be open, and now it's collecting reward, and it's going back to the start arm. And it's very well trained, so it knows what it's doing. And you see also actually this green light. This is actually fiber optics, where we can shine green light into the brain of the mouse. You will see later why this is important. And now this mouse knows what to do, and you see it goes to the right arm and collects the other reward. And now it can go back to the start arm, and then you start a new trial, and it can completely forget what it has done in this trial, but it still has to remember the rule and to incorporate the rule. So if you now inhibit the medial dorsal thalamus, you will see here. So now Sebastian Parnado, a postdoc in the lab, he used the mice, and then you can train the mice, so you're doing well the task, and then you start playing with the delay and increase the delay. And what you expect to see is basically, as long as the delay is more difficult as the task, and as worse the mice are performing. So here you see the correct choices in the task. You see they're pretty well at 10 seconds or 30 seconds delay here. But when you make it longer, 60, 90, or even 2 minutes, you see that any mouse will actually be worse in the performance. But if you now inhibit the medial dorsal thalamus, performance actually even gets worse. So that shows that the medial dorsal thalamus is important for this working memory task. So that also shows that it could theoretically originate, a deficit could originate in the structure. So then we teamed up with Pia Calzioni and Josh Gordon, and the hypothesis was, well, the medial dorsal thalamus leads to these deficits because you disrupt communications with the prefrontal cortex. And what you can do now is you record from both structures, from the medial dorsal thalamus and from the prefrontal cortex simultaneously. And if this circuit is used, then the synchronous activity between both structures should go up because they should fire together if they talk to each other. And that's actually what you see. If you now look, when the mice are learning the task, actually you see that over time, this synchronous activity here gets higher. So they're firing more and more together as better they're getting in the task. And moreover, during the choice phase, when they have to remember something, it's even higher than during the encoding phase when they're encoding the information. And both of this you disrupt when you inhibit the medial dorsal thalamus. So this is a good indication that the MD, the medial dorsal thalamus, may regulate this behavior via its projections to the prefrontal cortex. So we thought, okay, let's test this now directly by only inhibiting the projections from the medial dorsal thalamus to the prefrontal cortex and see whether this hypothesis is true. And this is Scott Balkan, a student in the lab, did this. And this is where now the green light is coming in. So I've shown you the mouse that we can shine green light into the brain. And what the green light is doing, it's actually activating what's called an inhibitory opsin. So this could be either an ion channel or a proton pump, which when you activate this with the green light, you can actually inhibit also these neurons. But now you only do it when the green light is on. And the beauty is actually that this opsin, when you express it here, it's actually transported to this axonal terminal, so the nerve ends of these neurons. And then when you activate it, it inhibits the synaptic transmission, which means that the medial dorsal thalamus neurons cannot talk to the prefrontal cortex anymore because you block the information from going there. So if you now put your fiber optics basically here in the medial prefrontal cortex, you can inhibit only this projection. And so when Scott did this, so he then asked the question, well, but is it really specific? The hypothesis was only if you inhibit projections to the medial prefrontal cortex, which is important for working memory, then you should see a deficit, but not if you inhibit any other projection, let's say, to the orbital frontal cortex. And that's what he did. So if you use a 10 seconds delay, like I have shown you before of the medial dorsal thalamic inhibition, you actually don't see much of a deficit. But if you increase the delay, first what you see that the mice in general, it's harder. So it's now, before there were 90% correct trials, now it goes down to 75% correct trials. But also if you now inhibit this projection, you see a deficit, but not if you inhibit the other projections. So that shows us that indeed, the medial dorsal thalamus regulates working memory via its projection to the prefrontal cortex. Now the beauty of the system now is this optogenetic system where you can light activate these channels. It has a very high temporal resolution. Now you can even ask, when do we need these projections? Do we need it here when the mouse has to encode the information? Do we need it here when the mouse has to remember something? Or do we need it here when the mouse has to use this information to solve the problem? And basically what we observed, or what Scott observed when he inhibited these projections, you need them during the delay or maintenance phase. So if this is the case, that would suggest that if you inhibit now the projections from the medial dorsal thalamus to the prefrontal cortex, that there should be some activity during the delay in the prefrontal cortex that you inhibit, and that can actually predict whether later in the choice phase, the mouse will do a correct or an incorrect trial. And so that we looked for, and the hypothesis was that the activity when the mouse is sitting in the start box during the delay phase, that you should find neurons that have a higher activity there than when the mouse is sitting in the same place later between trials. And this is actually the case. So you find neurons. This is a neuron, one neuron in the prefrontal cortex, yeah, that shows elevated activity during the delay, But importantly, you see that this neuron shows high activity early on. So this is a 60 seconds delay. Yeah? This shows high activity early on in a correct trial, but this activity collapses when it's an incorrect trial. And this you basically measure during the delay, even before the mouse has made the choice. Later in the choice, it will make a mistake or not. Here's another example of an innate neuron that shows high activity doing a correct trial, but low activity doing an incorrect trial. And then about 30% of prefrontal neurons are doing this, and you see they don't cover the entire 60%, but they kind of tile up the whole delay. And one idea is that they actually talk to each other during the delay. And then, importantly, doing incorrect trial, you see that this activity collapses. And then these neurons, these are the ones that are sensitive to when you inhibit now the inputs from the medial dorsal thalamus. So here you can cluster them. You see they're high doing correct, low doing incorrect. And now if you inhibit the projections from the medial dorsal thalamus to the prefrontal cortex, you see actually that this activity collapses. Interestingly, during the later phase of this delays and not early on, that may suggest why the mice are still good at doing it at 10 seconds, but not with 60 seconds. So we then reasoned if this, so basically what this shows is that you need the medial dorsal thalamus to keep the prefrontal cortex running during this working memory test, to keep the activity up there so that the prefrontal cortex can do its job and basically solve the problem. So if this is the case, we reasoned that if we now stimulate or excite the medial dorsal thalamus, maybe we're actually able to improve the working memory in this mice. And so we did this with another opsin where you can now increase the excitability of this neuron so that they're more easily activated, so that you boost the function, you make it stronger, basically. And when you do this during the sample phase, you don't see much of an effect because we know the circuitry is not important for encoding. But when you do this during the delay phase, you see now that the mice are getting better. So basically, the one thing I want you to, or the two things I want to remember from this talk is that the prefrontal cortex doesn't function on its own. You need the medial dorsal thalamic input for sustaining this enhanced cortical activity during a working memory task. And this is probably a general principle that may be also important for other prefrontal dependent cognitive tasks like attentional state shifting, for example. And then boosting the medial dorsal thalamus may be a strategy to enhance prefrontal cortex functions. And of course, the question is whether you could do something like this in patients. And with non-invasive methods, right now it's very hard to go down there in the middle to the thalamus. But people are developing really cool things like using strong electric fields where you then can bundle actually electrical activation non-invasively just to deep structures in the brain. So it's not completely crazy to think that you actually would be able to enhance the function of the medial dorsal thalamus. So at the end, I would like to thank the people that actually did the work. It was Scott Balkan and Sebastian, and then Pia with the recordings of the first project. And we had a wonderful collaboration with George Gordon. And Sarah Canetta actually did the study that Alan mentioned at the beginning, the maternal immune activation study.
Questions? Very interesting presentation. I would like to press you a little bit on the practical implications of your work, particularly the optogenetic inhibition. Can you see a time when that would be able to be translated into something actually clinical?
Well, I think with light may be hard, but so one idea is there was a really interesting study from Boyden Lab. It's actually one of the developers also of optogenetic, where they used electric stimulation. And it's basically, if you use a very high frequency, this would not activate your neurons. And then if you take, and you, let's say, shine from the light side, and then you shine from the other side with another very high frequency, and then you just have a difference, let's say, of 10 hertz, yeah? Then what happens, actually, you get kind of an envelope activity only at 10 hertz. And this is a frequency that can activate neurons. So you could imagine to have like one here, it goes to the medial or the thalamus, and then, don't know, whenever you have to concentrate or do something important, you switch the thing on, and it may help you. I mean, this is science fiction, but it's not so far out, actually. Really. And the other thing that people are working is with the viral methods we use, where we can express proteins only, let's say, in the thalamus, like this designer receptor. People are working now on viruses that you can give systemically, that then they cross the blood-brain barrier. And then when you have specific promoters, you could imagine to have specific expression only in the cells you want. And then you could inject a designer drug or find other ways to activate them. Light is hard to get so deep, but who knows, yeah.
Well, with those kinds of injections, is there any way to get to one side of the brain versus the other? I mean, would you be able to localize?
Well, not with the systemic, unless your gene, your promoter is lateralized. Yeah.
Okay. Are there other questions for? Oh, yes. I'm sorry.
Thank you. Really nice work. The mice that you're working on are actually healthy mice. They have a completely well-functioning thalamus before you made your intervention. Is that correct? Now, if the thalamus is actually damaged, as it's the case in schizophrenia, what do you expect your manipulations might have as an outcome?
Yeah, well, that depends how exactly the damage is. So my hope would be that there would be still enough projections to the prefrontal cortex, and I think that would be the case. And so that if you stimulate them, that you may still get the effects that we see here in the wild-type condition. Yeah. But that's a good question. I mean, we know there are structural imaging findings suggesting that it may be smaller. There's some post-mortem findings that there may be less neurons, but it's not completely wiped out. So I think there's enough thalamus left projecting up there so that if you would activate the system, that it still could have an impact, yeah.
Where you want them to go, but you might be activating other pathways that you do not wish to.
Yeah,
I think that's the
concern. It's a possibility. If you have this, then it gets more complicated, yeah.
Okay, all the way over here, sir. If you could, yeah, okay.
If ECT could be used in any way to activate the thalamus, I don't know if it does, and therefore, even if it's not direct, it could have some effect here on the medial thalamus area.
No, I think ECT, my understanding, it's getting at the whole brain. So that would be, I think you induce seizures. So that would be not the specificity we want to have. Then transmagnetic stimulation, it's hard to get so deep. So we need some improvement on techniques that can go to the deep structures. Yeah.
Okay. So either way.
There are other conditions where working memory is deficient, correct? And is this model applied to those conditions?
Yes, so I mean this, as we discussed before, these are even normal mice. So you could imagine that this could happen in any condition where these projections are preserved to improve the working memory. So it's not necessarily specific for schizophrenia. We just use schizophrenia as a starting point based on the imaging finding, but working memory is definitely not only affected in schizophrenia, yes.
I just want to mention that ultrasound may be a technique that can find and stimulate very small areas anywhere in the brain. It's now used in Parkinson's and essential tremor because it does actually damage the cells, but it could, at a lower power, potentially be used safely to stimulate or inhibit very specific areas. So there may be a human application to it if we can ever show the safety of it in terms of not damaging the neurons that are stimulated.
Yeah. That was a comment, no? There was, yeah. No, but it's good, yes.
Does this have implications for, say, the treatment of Alzheimer's patients?
That's a good point.
Repeat the question, because she might not have.
Yes. No, I think it could, but I don't know. I don't know enough about the Alzheimer literature with regard to how the thalamus is affected. But theoretically, you would think whenever I improve PFC function, that should be helpful. I mean, that would be my answer to the argument.
Thank you very much, Dr. Don Both.